How does the virus, like coronavirus, actually manages to completely take over a cell that it infects? It's a tiny little thing that gets inside a huge cell, and yet it completely hijacks it from its normal functions and uses it for its own purposes. And then, how does melatonin might protect from that? My name is Dr. Michael Arashik of Neurogenomics. We're going to discuss second part of the video related to melatonin protection from COVID-19 complications. This is based on the same review that I used last time as well. And today we're gonna hit a twilight zone when it comes to what the SARS-CoV-2 virus can do. All right, so let's start. So normally we always focus about the spike protein, right? Well, obviously the virus produces many other viral proteins as well. And the one that I want to focus on right now at this particular moment is one called nucleocapsid protein, also abbreviated as N protein. So you know how we abbreviated spike protein as S protein. You can abbreviate nucleocapsid protein as N protein. And that particular protein has an interesting function. It can create, it helps to assemble what is referred to as viral factories. Now that's not an official name. Basically, what it does, it creates this unusual territory where the liquid becomes denser. It's more gel-like. And the official name of that is called liquid-liquid phase separation. So it almost becomes like a separate organelle, but it's without any membrane. It's not surrounded by any membranes. Now, recall organelles are basically different components inside the cell that would be like mimicking our own organs inside our body. So a good example of that is mitochondria, which we're going to be talking about in a moment. It does its specific function for the cell, or nucleus is another example, but they are surrounded by a membrane. Now, this liquid-liquid phase separation that becomes denser gel-like entity, there is no membrane, and yet it has its own demarcation zone. And nucleocapsid protein can achieve that by interacting with certain cellular molecules, including cellular mRNAs, as well as some of its own viral RNAs. Now, nucleocapsid part of it apparently can become I was gonna say disoriented, disorganized. So what it means, it doesn't actually have a proper three-dimensional structure. So remember, when proteins are being made from mRNA template, mRNA is just a blueprint that is being used in order to actually produce the protein. Then when you produce the protein, you assemble chains of amino acids in a single long chain. Now, amino acids, of course, there's many different kinds. They're made out of different atoms, and all of them have a specific shape, specific size, and also specific electric charge. And as a consequence, because of all of these factors, these amino acids can start interacting with each other in a variety of different ways and help the protein assemble into a three-dimensional entity. So then the protein actually gains a three-dimensional structure, okay? So, but if it's disorganized, then it's just a linear chain of amino acids that can flap around. And what that means at that point is that all of the amino acids along that chain, they're free to interact electrostatically with anything else around. And the nucleocapsid proteins apparently have these unassembled components that help to produce this liquid-liquid phase separation. Now I'm going to pause on that for a moment, but we are going to come back to this in a moment, briefly. And right now I wanted to now talk about another viral protein, which is quite a doozy. This is a bit of a monster. So again, everyone thinks spike protein is bad, and it is. But here's another nasty little guy that that virus can produce, and that's called an envelope protein. So what does the envelope protein do? Also abbreviated as an E protein. Okay, so what does it do? It creates a channel. It creates a channel that can disrupt the integrity of the cell that infects. So how does it work? E protein is a homopentamer. Homo means that it likes to, in this context, it means it likes to hang out with itself, with its own kind. And pentamer means there's 5 of them. So that means 5 identical envelope proteins come together to assemble and form a channel. It's just like spike protein is a homotrimer. It means that the spike protein, whenever we talk about spike protein this, spike protein that, it's actually not one single spike protein, it's 3 identical spike proteins coming together to form the final 3-dimensional structure that resides on the surface of the coronavirus. And this is what we're studying all the time. So whenever we're talking about the spike protein, in reality, we are actually talking about three proteins that come together. Okay, so this envelope protein, homopentamer, creates a channel, and this channel can basically perforate the cell, and as a consequence, it disrupts the proper function of the cell. Literally, things can now diffuse in and out from the cell in an uncontrolled manner. Not what you want. This is eventually disrupts the cell and will lead to cell death. So that's how viruses can hijack the function of the cell. So this has a lot of consequences to the cell. So let's talk about that right now. So you can see how bad the E protein is. So number 1, the biggest one that we're going to mention is that mitochondria is disorganized. Recall from the past, mitochondria are the tiny little organelles inside cells that are responsible for producing ATP. So they are basically energy factories. ATP are molecules that can be used as a storage of energy that then cell uses in order to really exist and function. We absolutely need ATP to exist. We need its production all the time, constantly. That's what we need for energy. So think of ATP as a coal that you dig out from the ground in order to convert it into energy. So the same thing works for ATP, and mitochondria is responsible for all of that production. Okay, so then if you start disrupting mitochondria, then you will disrupt the proper production of ATP. So how does it work? Number 1, you actually depolarize the membrane of mitochondria. So what does that mean? It means that previously you would have specific charge assembled on inside and the outside of the membrane of mitochondria. And when you depolarize that, it means that you have altered that electric charge, which is necessary for proper functioning of mitochondria. We talked about that in a previous video dedicated to melatonin as well. I'm standing on an icy slope because I'm on a glacier basically, and there's, as you can see, there's ice all around me. It's so beautiful. And another thing that this does to the mitochondria is that it actually also changes its shape. So the mitochondria starts swelling, and basically its shape changes. And as a consequence of these 2 events, the depolarization and change of its shape, mitochondria changes its proper function, and it no longer produces ATP through use of oxygen. This is why we need to breathe. This is why we need intake of oxygen. Mitochondria uses oxygen to produce ATP through a process called oxidative phosphorylation. The name is not that important. And as a consequence, the mitochondria in its disrupted state produces ATP without oxygen, but it means it now produces it in much lower quantity. And this also helps in the development of that nucleocapsid protein promoted liquid-liquid phase separation, or in other words, formation of those viral factories. And those viral factories, by the way, this is where the virus is then going to be duplicated and assembled to form new progeny. So this is basically where you start copying the material and so on. So that is one of the events that happens as a consequence of the proper disruption of the cell because of the virus inserted those envelope protein ionic channels. Okay, there's other events that also happen. So as a consequence, you increase the oxidative reactive oxidative stress through the reactive oxygen species as well as reactive nitrogen species. We again also talked about that previously, and the consequence of that is, of course, these are very damaging. They can be damaging to the mitochondrial DNA, and when you damage mitochondrial DNA, you will force the cell to commit suicide, and this process is called apoptosis, aka by the way, I should mention, we have tiny amount of DNA inside the mitochondria, but the vast majority of all of your DNA you're born with resides inside the nucleus. But there's still a super tiny amount of DNA that mitochondria has, and it actually is responsible for production of some of those mitochondrial proteins as well. And the cool part is that the only way, as far as we know at the moment, is you can inherit that DNA from your mom. So it's only passed on from mom to all of its offspring. And this way you can actually track inheritance. But there was some information published, I believe this might have been last year. Someone suggested and showed some evidence that sperm could carry mitochondria as well. But this is obviously not accepted at the moment because there was only one such information published, and there could be other reasons why this might have been observed. There's simply not enough evidence to believe that the sperm could be passing on mitochondria. Right now, it's believed that you only get mitochondria from mom. That's basically from the fertilized egg when sperm and egg combine. Okay, so that's extra little bit of information right there. But this is basically oxidative stress is not a good thing, not what we want normally, and we do normally want to protect ourselves from that as well. And then finally, you can also start changing the lipid molecules as well. Those are fat molecules that, for example, can be used to make up the membranes of the organelles or the cell, and that also, all of that together can also result in even further and faster depolarization of the mitochondria as well. So it's like a compounding effect. Basically, mitochondria is in trouble. Okay, so there's that. But there's other things that also happen as a consequence of this envelope protein. And by the way, I'm gonna get to melatonin eventually. You just have to bear with me and maybe just enjoy the scenery in the meantime. So what else happens there? So this production is so cool. The glacier, you can hear a little bit of melting here and there, and the rocks are coming down. It's really neat. So what else happens is it promotes the development of that nucleocapsid viral factories, this liquid-liquid phase separation I just mentioned as well. And it does that through, and I apologize for the wind, and it does that, in order for that to happen, you need to steal a protein, an important protein from the cell. This protein is called, I believe it's called DDX3X. And what's interesting is that this particular protein also plays a crucial role in the formation of stress granules. Now, what are stress granules? These are another gel-like organelles without membrane. So very similar to those viral factories. It's another way that you produce this liquid phase separation, where you have this denser fragment inside the cell than the rest of the cell. And these stress granules are formed during cellular stress, such as viral infection. And what do? They're supposed to help you fight the viral infection. So one of the ways you do that is by promoting activation of some of the antiviral responses, including interferon response. And that's through this DDX3X protein. So when you produce the viral factories, you steal that protein away from stress granules. So stress granules no longer do their proper, they no longer can be formed properly. And finally, on top of all of this, if this wasn't enough, there's one more thing that I will mention, and this is that these viral envelope proteins that form these channels as a consequence of causing the stress to the cell, it also helps to induce the formation of inflammasome. I believe this is NLRP3 inflammasomes. I just mentioned this once in my series of videos related to shedding. And again, those inflammasomes require that DDX3X3 protein as well for its formation. So you can see the virus achieves multiple things in one go. First of all, it helps to create viral factories, and then also it removes the formation of stress granules that would be helping the cell, and finally also promotes the formation of inflammasomes, which eventually will also lead to the death of the cell, because formation of inflammasomes also promotes apoptosis, which is basically controlled suicide of the cell. So, and together, all of this means that actually this envelope protein of the virus can be pathogenic. So it can promote evolution of the pathogenicity of the virus, because if you think about it, the better this envelope channel becomes that disrupts the cell, and the better you can destroy the cell, the faster the virus will be able to shed its progeny, and therefore, hence you can see it's more pathogenic. And indeed, this has been observed in the past, where certain mutations, remember, mutations arise randomly all the time, every time genetic material is being duplicated, and certain mutations in the envelope protein were correlated with increased severity of COVID-19 disease in some of the patients. All right, so that's about that, and how the virus can just completely hijack the cell. So where does the melatonin come into this? Well, first of all, melatonin, you cute little wonder molecule. First of all, it helps to restore proper mitochondria function. So then it ensures that the mitochondria remains normal, if you will. And on top of that, it also reduces the formation of those viral factories. And it achieves that in a couple ways. So number one, it actually leads to phosphorylation of the nucleocapsid protein. It promotes phosphorylation of the nucleocapsid protein. So what is phosphorylation? It's basically you plop on a phosphate atom molecule on top of a protein. And that's one of the most common on and off switches in biology. So basically, and where does the phosphate come from? I wonder if you guessed it, ATP. So there are proteins that actually put on phosphate on another protein. These are called kinases. And the ones that remove phosphate from proteins, these are called phosphatases. And this is a very common way of making or asking a protein to change its function, because once you plop a phosphate molecule in a the right strategic location on a protein, the protein will change its activity, because suddenly you change both its three-dimensional structure by putting that extra phosphate atom, as well as its overall local electric charge. Okay, so, and that can have profound effect on what the protein can do afterwards. So it's a common on-off switch, and in this case, melatonin promotes phosphorylation of nucleocapsid protein, which then reduces its likelihood in formation of those viral factories. Okay, so that was super interesting. And then finally, also melatonin helps to restore those antiviral activities by making sure that DDX3X protein can continue performing its function, which is to activate the antiviral genes, including the interferon pathway. It's one of the important innate immunity cytokines that helps to fight the virus. So overall, what this means is that proper use of melatonin early on in infection can be very, very, very useful in helping you deal with infection. So something that I wanted to share, because it was just simply too much to be able to tell you all of this based on that review in the last video I made on melatonin. Definitely an incredible amount of information. The only thing that I didn't like about that review is that sometimes I did not see references for some of the claims that were being made, or at least I could not correlate that. But clearly the authors of that review know a whole lot more than I will ever be able to scratch the surface on this topic. So we're just going to go for now with their word, and we'll see what else we can learn in the future. That's all I have for you right now. I hope you like the scenery. And if you like the scenery and the topic on top of this, then please give me a like, share the video. Obviously, this is how we grow. The more supporters we have, the bigger the channel, and that's good for a variety of reasons. Thank you for the donations as well. I always keep forgetting to mention that, but they're deeply appreciated. I obviously use that money as well to hire help for video editing, as well as dealing with some of the questions, because there's so many. It's simply impossible to deal with all of this. And please keep adding comments and questions. And the last thing I'll mention is we obviously have a series of COVID Q&A events. And if you're interested in those, you can and you would like to try getting it for free to start with, then we'll send you a free ticket if you subscribe to our newsletter. It's a venue where you can help to shape as well, to a degree, how we're presenting science, because melatonin, boom, it came from that. And in the end, I loved it. Once I studied it, I was like, wow, this is an incredible compound. So hopefully I'll see some of you there, and I'll see you in the next installment. Till then, hey, go outdoors as much as you can, stay active, stay healthy. All the best. Bye, everyone.